right. Hello again, everybody. Not sure, we're guessing most of you were here before, but welcome to those who weren't. My name is Jim Way. I'm Executive Director of the American Astronautical Society, your hosts for these technical sessions for the ISS R&D conference. We wish we could be in person, but such is life. We're here virtually, and we're just trying to make sure that we present a lot of this amazing work that all of these presenters have done. Yeah, welcome to the last segment of these virtual technical sessions. This one is fundamental biology. And so without further ado, we're just going to get right into it. And I'm going to turn it over to Ryan Reeves. Ryan, Ryan is the Technical Director for Research and Innovation at the ISS National Labs. Ryan, take it away.
All right. Well, thank you very much, Jim. Happy to have been part of and see all the great presentations that we've had so far the past couple of days. This will be the last session that we have. So this is fundamental biology. We have 4 talks today. So for the process of this, we will present the 5-minute executive summary, and then we'll open it up from questions from that author. We'll then move on to the next presentation and so forth. After each presentation, we'll have the author come on screen and begin the Q&A from anyone here on the webinar. For those of you who are attending, you won't be able to come on screen if you have a question, so please use the Q&A tab down below in your Zoom link. And we'll ask those questions in real time. If we have any additional time at the end, we can answer additional questions. If you are interested in the full presentation, the full 20-minute presentations, please visit the online program and open up that file. Jim has now just put that into the chat. So with that, we'll start with our first presentation by Jonathan Galaska of NASA Ames Research Center. So we'll start that presentation now.
Hello,
Yeah, it
my
looks
name is
like it.
Jonathan Galaska. I'm the acting OSDR project manager, and I'm happy to tell you about our efforts towards building an open science ecosystem for life in space. The Space Biology program in the Biological and Physical Sciences Division of the NASA Science Mission Directorate uses the space environment to advance our knowledge of how gravity, radiation, and other spaceflight stressors affect living organisms. Decades of research on various platforms has produced a treasure trove of data on how diverse model systems respond to the space environment. NASA has developed an ecosystem of open science tools to maximize the value of these datasets and accelerate the pace of discovery to support the NASA mission. The Open Science Data Repository, or OSDR, sits at the center of this ecosystem. It contains omics data, physiological data, and biospecimens from experiments investigating the impact of spaceflight on biological systems. It consists of a suite of tools including GeneLab, ALSDA, EDA, RadLab, NBISC, and SMCC, which together allow users to contribute, discover, and explore data. In the past decade, the amount and diversity of data available within OSDR has continually increased. OSDR contains 580 studies featuring 1,029 datasets. It contains data from 45 different species and greater than 90 different assays. While much of this data is from NASA-funded studies, it also includes studies funded by various international space agencies. To foster a community dedicated to analysis of OSDR data, we have developed a set of OSDR analysis working groups. These groups self-organize into subgroups dedicated to specific domains of interest. These groups serve 2 functions. First, they provide feedback on our data systems and processes. And second, they generate novel discoveries and accelerate progress on the NASA mission. The impact of the OSDR infrastructure and the analysis working groups is well illustrated in a recent publication. The analyses within this publication were motivated by the fact that feeding astronauts on long-duration missions is a major challenge. In fact, this challenge has been formally recognized within the Human Research Program's Food and Nutrition Risk and the Moon to Mars Architecture Gap 0305. In response, consumption of fresh vegetables is being considered as a method to mitigate risks and bridge these gaps. This publication resulted from a unique collaboration between 3 analysis working groups, the plant, human, and ALSDA working groups. These groups came together to analyze 9 separate data sets within OSDR, including data on the nutritional content of plants and the physiological responses of astronauts to spaceflight. They were able to document differences in the nutritional content in some space-grown crops, including a reduction in calcium content. Moreover, they were able to connect this to disruptions in astronaut calcium signaling pathways. Overall, this serves as a roadmap for how integrated analysis of data can improve nutritional planning and enable exploration. Thank you for your time and attention, and I'd also like to thank my co-authors on this presentation, the entire OSDR team for all of their efforts, the analysis working groups for their continued dedication to this project, and the funders who make all of this possible.
Okay, well, great. So thank you very much for that presentation. Again, if you have any questions, please enter them in the Q&A tab below, and we'll field those now. First question, what calculations have been performed to determine how much food can be taken or grown in flight to distant planets?
Great question. A lot of calculations. I'm not particularly familiar with that type of like logistical mission planning. It's certainly been looked at in great detail. And there's sort of 2 sides to that. One is just providing bulk calories, enough energy, protein, macronutrients to the crew. But there's another side to it, which is providing micronutrients. So labile vitamins. And that is where we think that crops are going to come into the picture. So you may not need a large amount of crops in those scenarios, but providing those micronutrients is an area where a living system may be very important to effectively produce those micronutrients on demand, which is what a plant growth system can do. And so an important question there, of course, is what is going to be the nutritional profile of crops grown in space? And so that publication really was looking at that and specifically connecting that to the status of the astronaut. So the ability of an astronaut to absorb and utilize nutrients, but also the needs of an astronaut for a particular nutrient profile could be different. And so that publication was really looking at that integrated analysis, both what can the plants provide, but also what do the astronauts need, and how will the astronauts be able to utilize those micronutrients. So good question. There's a lot of research in that regard. I'm just not that familiar with those logistics. Thank you.
Sure. How might OSDR help close the Moon to Mars gap? Are there any specific pathways that you're aware of that kind of exist in the database now?
So that specific gap is really pointing out this issue with food. I mean, I think that there's a huge opportunity there. I think looking at all of the plant research that exists within OSDR and trying to understand how plants are growing in space, that is really the strength of OSDR and BPS research, is providing that fundamental knowledge on what is happening to these systems. So I think that's where we really want the community to jump in. The investigations that we've curated are typically targeted towards a specific hypothesis, which they generally answer very well. But our philosophy is that many other questions can be answered about plant growth, about the potential for those plant species in that hardware to provide a specific service to the astronauts. So again, great opportunity, and one of the things we like to do is rely on through these working groups is rely on the creativity of the community to really answer the questions that we or the PIs weren't actually even considering when they designed those experiments.
Okay. So according to the question, vitamin B, B vitamins are not available from plant-type foods. What are the plans for those type of nutrients?
Great question. Again, I am not a nutritionist and don't know the details, but you know, there are lots of concepts for how to provide these sort of labile nutrients. One, of course, is just developing better storage um, infrastructure, so to make sure that stored labile nutrients remain intact. But plants are an option, but also microbial production is being considered. There's an interesting experiment called bionutrients, looking at the ability of baker's yeast to generate carotenoids. So I think there's many routes to potentially providing these that we want to investigate. Again, from our perspective, the ability to produce these on demand with a living system, a living bioreactor, whether that's a plant or a microbe, or a fungus, is of particular interest in something that BPS, through some of its newly defined goals, will look to investigate moving forward.
Okay. And maybe last couple questions, I'll kind of group these together a little bit. So are the results in the database, are they primarily for ISS? Do you include suborbital or other, flight opportunities like programs? And if there are PIs out there that, are say outside of BPS, but would like to contribute their data and experiments, how might they go about that?
By volume, most of our data does come from ISS experiments. That's just a reflection of spaceflight research. But we are platform agnostic. So we look for any, we of course are focused on BPS funded experiments, but we reach out pretty broadly to bring in spaceflight relevant biological experiments. So no, it's not limited to ISS. And as we move to new platforms, we're going to be looking to pull in data from those as well. So we do look broadly. We try and provide an integrated set of data that can answer broad questions. And we always welcome contributions. So you can reach out to myself or the team. If you go to the site, we do have some pointers on how to get in touch with us. And we have a set of curators who will work with you to get your data uploaded.
Okay. Well, thank you very much. We appreciate your talk and your time in answering questions. All right. Our next talk is going to be by Benata Jadar, sorry, from Oregon State University. So we'll play that next executive summary here.
It's the narration for the project title, Modeling Cardiac Atrophy Utilizing Engineered Tissue Platforms by Subjecting Them to Microgravity aboard the International Space Station National Laboratory. The project was authored by Dr. Benata Jadar with myself, Ivana Hernandez, as the first author. This work was carried out at Dr. Jadar's IMSTEL lab at Oregon State University. To begin with, the main hypothesis of this research is that exposure to microgravity may induce measurable changes in cardiac-specific biomarkers, cell morphology, and cell-to-cell interactions within engineered human cardiac organoids, thereby mimicking the phenotypic characteristics of cardiac atrophy. To accomplish this, we developed and characterized electrospoon polycaprolactone scaffolds coated with matrigel for cardiac cell culture. SEM analysis showed increased fiber thickness and reduced porosity with matrigel coating. Contact angle measurements confirmed improved hydrophilicity, while FTIR data revealed the presence of collagen and laminin, confirming successful matrigel incorporation. These outcomes prove the suitability of the PCL matrigel scaffolds for cell culture, as evidenced by SEM showing AC16 cardiomyocytes adhere to the scaffold and an increased DAPI count indicating enhanced cell attachment and proliferation. After developing PCL matrigel scaffolds, cardiac organoids were engineered by seeding human AC16 cardiomyocytes, cardiac fibroblasts, and microvascular endothelial cells on top. These organoids were exposed to three conditions: ground controls, including 2D cultures and 3D scaffolds under Earth's gravity; simulated microgravity with 3D scaffolds in a rotating bioreactor; and real microgravity using 3D scaffolds aboard the International Space Station, housed by Space Tango's Cube Lab. This presentation focuses on cardiomyocyte gap junction expression and oxylipin concentrations, comparing Earth's gravity and real microgravity, highlighted by the dotted blue arrow and boxes in this slide. This section shows immunostaining results for connexin 43 in cardiomyocytes cultured under 3D ground control and 3D real microgravity at 7 and 21 days, along with quantification per field of view. A time-dependent decrease in connexin 43 expression under real microgravity indicates disrupted gap junction integrity. Quantitative analysis further confirms reduced connexin 43 levels compared to ground controls, suggesting development of cardiac atrophy. Shown here are oxylipin concentrations, derived from polyunsaturated fatty acids quantified via liquid chromatography mass spectrometry. The data includes a heat map of all conditions, post-flight comparisons at 7 and 21 days in cardiomyocyte monocultures, and a similar comparison in co-cultures of cardiomyocytes, fibroblasts, and endothelial cells. These LCMS data highlight distinct oxylipin signaling profiles influenced by both cell culture composition and duration of microgravity exposure, suggesting differential inflammatory and metabolic responses under spaceflight conditions. In conclusion, the PCL substrate was successfully modified with Matrigel to improve cell adhesion and hydrophilicity. PCL Matrigel scaffolds were successfully developed and demonstrated the ability to withstand extreme environments for longevity studies. Changes in connexin 43 gap junction expression under real microgravity suggests early cardiac atrophy-related remodeling. Oxylipin signaling profiles suggest that both the cellular context and the duration of microgravity exposure play critical roles in shaping the biochemical landscape of cardiac tissue. Ongoing RNA sequencing and pathway analysis will deepen understanding of signaling mechanisms. Microgravity serves as a valuable platform for studying the underlying processes of cardiac atrophy, with important implications for understanding cardiovascular dysfunction. The insights gained from this research may inform the development of effective countermeasures to reduce cardiovascular risks in astronauts as well as patients on Earth.
Okay, great. Well, thank you very much. We have some questions, but again, please feel free, go to the Q&A tab and enter your questions there. So our first question is, did your cultures of these cardiac myocytes begin spontaneous contraction? What additional influences did the endothelial cells provide?
Yeah, that's a great question. And thank you, Russell. So these cells, the AC16 cardiomyocytes, these are derived from the adult human ventricular region. These are genetically modified to not spontaneously contract, and that way these are not terminally differentiated cells. You can keep passaging them and repeating them in several experiments. So that hopefully answers the first part of your question. I will tell you though that we did measure calcium transients and we do see interesting trends which vary between Earth studies and simulated and real microgravity, and those are significantly different. So watch for all that as we wrap up our publication, and hopefully that's something interesting to look out for. To answer the second part of the question, what influences did the endothelial cells provide? They just provide some paracrine signaling for the cardiomyocytes to behave in a more healthy manner. So in cultures where we had just cardiomyocytes versus in cultures where we had endothelial cells added to those cultures, the inflammatory signal was much lessened and the impact of microgravity was much lessened. hopefully that answers the first question totally. The second question, um, that is something that, yeah, when we try and publish this work, we've done some other work on suborbital flights before. This is also a question that we get commonly from reviewers. High G forces, it's very difficult and challenging for us to dissect just the effect of launch and vibrations during, the initial phase of the flight. So we just say that, yes, but yeah, we're trying to brainstorm or finding a partner who can help us dissect out those two phenomena. So I don't have an answer for you yet. I think I lost track. Did you measure any cytokine production with the endothelial cells? Yes, we're doing that now. The oxylipin data that we showed you gave us some insight into this. So we did see some initial cytokine production with the CD31 marker, and we're probing more into that with our RNA-seq.
Okay. So I guess, if you were to have follow-up studies, where would you go next with this?
We are planning, we are seeking funding from cases as we speak. And in the past, this was a cardiac system, and we've also published with a neuronal system. And our goal is to do a combined cardiac neuronal tissue model, something that could better mimic stroke. So that's something that we need help either from the cases or ISS national labs or even NASA solicitation. But yeah, that's what would be our next step.
Okay. And last question, are the cells shielded from radiation effects? Do you see any effects of radiation?
So we are being told by our space implementation hardware partner that all the rockets and the space engines, they do have the necessary shielding. We're typically trying to also dissect out those effects. So we are currently doing Earth studies where we expose ourselves to radiation and see if those responses were different from the ones obtained on spaceflight. And we have electrophysiology measurements also trying to see, if the cardiac beating will differ on spaceflight versus Earth controlled. So yeah, those are some things we're probing along as we move forward. But yeah, it's a lot of work to get to those answers.
Okay. Well, thank you very much for your presentation and for your thoughtful answers. We very much appreciate that. Now going on to our third speaker, we have Matsufumi Maritomi from the University of Tsukuba. So we'll play their presentation now.
Hello, this presentation is prepared by Masafumi Muratani, Agatha Rudolph, and Alexa Sadia, titled Space-Induced Atavistic Phenotypes Observed Aboard the ISS highlights the impact of continuous physiological adaptation in shaping the form of life on Earth and beyond. This slide shows a collection of JAXA space experiments on ISS, including worm, fish, mouse, and human. During the course of our integrative analysis, we noticed that potential link between space phenotype and evolution. This slide shows how vertebrates acquired different functions during evolution and which of them are affected in space. Basic systems like smooth muscle, cartilage, natural immunity were maintained, but in space, more specialized functions such as muscle fiber types, vestibular system, hard bones and bone marrow, as well as immune receptor diversity were particularly affected. These findings highlight that while basic functions remain intact, advanced functions acquired during the transition from sea to land are especially sensitive to the space environment. This slide highlights that common space phenotypes are not just the result of mechanical unloading. Of course, we see muscle atrophy and bone density loss, but there are also non-mechanical changes, shifts in muscle fiber types, reduced thymus mass, metabolic alterations, oxidative stress response pathway change, and circadian rhythm disruption, even in intergenerational effects. On the right, you see the context. These are functions that the vertebrates gained during the adaptation from sea to land: bone to support the body, muscles for locomotion, mineral and immune homeostasis, and stress responses such as circadian rhythm and UV protection. In space, many of these functions appear to regress almost like a throwback or atavistic phenotype. In other words, millions of years of evolutionary effort can suddenly be compromised under space conditions. To study these events further, we recently funded a 5-year grant from Japanese funding agency JSPS. For the rest of the presentation, I would like to explain the main theme of this study. On Earth, our body maintains homeostasis, keeping internal conditions stable despite environmental changes. For example, heat is produced or released to keep body temperature around 37 Celsius degree. This robustness ensures the basic body functions remain constant. In space, however, we observe strong plasticity. Bone density, muscle, immune function, and skin barriers all change significantly. This raises a key question: are these true adaptations or are they maladaptations, responses that compromise long-term health? To understand the future of life in space, we must consider both robustness and plasticity. Space environments affect genes, epigenetics, and developmental process across generations, leading to compound effects. Extending these studies to diverse species will help us to model how terrestrial life, including humans, might adapt beyond Earth. So this umbrella funding covers microbes, plants, animals, and human studies with diverse approaches for the exploration of environmental responses and robustness of biological systems to predict the future of life on and beyond Earth. Now we are looking for more affiliated projects and collaborators, so if you are interested in, please contact us. Thank you.
All right. Well, thank you very much for that. We appreciate the presentation. I believe our first question, I'm not going to be able to read myself. Matsufumi, are you able to see that?
Yes.
Or maybe that's the translation and it's just saying that
Yes.
it appreciates the conversation, the presentation. So, all right, I guess I'll ask a question, and please forgive my ignorance. The biology is not really my my field of study. But so my understanding is much of the changes that we see are in the expressions of genes. So if we were to look at, the reproduction and, further generations of that, how much do we see the, actual progression of any changes from one generation to the next? Maybe microbial would be the best, example of that. Maybe I don't know if that was a question or if you got anything from that, but
Yes, there is microbe observations are like changing. The one cell is a one generation, so they can quickly evolve or change. And one of our the mouse JAXA mouse flight indicated the
we did the in vitro fertilization of the sperm obtained from the further experiences space flight. And then that phenotype is affected to the next generation's phenotype. So unlike the really the genetically encoded change, there is some sort of intergenerational effects can be transmitted on even just one generation. So that indicates the Earth's environment is very, for example, gravity is very difficult to remove, but once we remove the big component, there could be very unexpected changes could happen.
Okay, thank you. So question here from the chat, are throwback adaptation gender or age specific?
Yes. So our observation was a mouse sperm experiment. The throwback is a general, I think. It's just a general phenotypic observation. The bone, muscle, and also other functions acquired during the sea to land transition is a kind of like a comprehensively weakened or lost in space.
Okay. While we wait for any additional questions, so can you talk a little bit about the next experiment that you're coming up and where you see this headed in the future?
We really want to see the multiple generation, like complete generation of a kind of evolution trajectory experiment. And obviously the mouse is a bit difficult. So we are also looking for the worm or fruit fly, or actually the plants experiment is plants field is really advanced in terms of the intergenerational or genetic change research. So we want to learn more from the plant world to plan better experiment for animals too.
Okay.
If we have opportunities.
All right. So next question. These primal systems in humans that are affected by microgravity, could it be that the neural systems connected to the primal systems in the brain are hardwired, therefore they are resistant to relearning?
For example, vestibular system, they seem to have a quite nice plasticity to adjust the body function to the space environment. So maybe the nervous system might be have like a different way to adapt or like adjust their function. But there also maybe there is a critical period during the development to adjust the function. Maybe that's the answer for neuronal part. There is a central control and also some cell-based cellular level of the gravity response and also radiation responses, I think.
Okay.
So it's
And then,
complex.
okay, thank you. And then the last question, with upcoming technologies that might enable magnetic shielding of radiation or artificial gravity, do you think that those would be able to address some of the biological problems that we might be seeing now, with weightlessness and exposure to space?
I think so, but it turned out to be gravity is very difficult to shield or change. So that is a very critical point. So the JAXA spaceflight experiment also tested the kind of like a moon gravity, Mars gravity effect, and it's under analysis. But this information is quite critical to design the future Mars and moon colony too, I think.
Okay. Well, thank you very much. It looks like there might be one additional question. So, Matsufumi, if you would like to stay on and answer that in the chat, that would be great. But we very much appreciate you. I'm not sure exactly what time it is where you are, but I believe it's not quite,
1 a.m.
Yes.
1 a.m. Yeah.
So
Thank you for
very much.
staying up with us.
Yes, thank you for your time. We appreciate it.
Thank you very much.
All right, so our last presentation is by Margaret Hitt from Columbia University. So we'll listen to her presentation now.
Hi everyone, thank you for being here. My name is Margaret Hitt, and today I will be presenting my research on melan-based substrate bioengineering for ISS and Artemis, optimizing nutrient uptake in NASA's Exudorf Pak Choi. So a little bit about me. I'm a sophomore at Columbia University studying biomedical engineering, and my research interests focus on space medicine and biology. Since high school, I've worked on space botany, and at Columbia I studied exosome-based therapeutics for astronaut health. Today's presentation comes from a project I began under Dr. Ramondas Cordero at Johns Hopkins University, exploring whether melanin, a radiation-absorbing biopolymer, can also improve plant growth in space. As space agencies plan missions to the Moon and Mars, astronauts will need fresh, nutrient-rich crops, not just packaged foods. But food grown in space is limited by substrate chemistry, especially nutrient balances like too much iron or too little calcium, magnesium, or potassium. That's where melanin may help. Beyond shielding radiation, melanin binds to iron, buffers pH, and retains moisture, which are all traits that could improve space agriculture. We tested Exudorf Pak Choi in two space-related substrates, arsenite and perlite vermiculite. Each was grown with or without melanin for 42 days in a NASA veggie-style chamber, as you can see on the right. Growth traits, substrate chemistry, and nutrient uptake were analyzed. So let's look over the results. Melanin clearly reshaped root zone chemistry, but in different ways for each substrate. In perlite vermiculite, it buffered acidity, tripled nutrient retention capacity, and boosted potassium and magnesium availability. In arsenite, it moderated alkalinity, but it reinforced iron and calcium dominance, which can suppress other nutrients. Overall, melanin didn't act uniformly. It tailored each substrate's chemistry in unique ways, showing its potential as a flexible amendment for space farming. Second, in terms of nutrient uptake, melanin reduced overall efficiency, likely because nutrients stayed bound in the substrate. But in arsenite, melanin promoted higher capture of phosphorus, potassium, magnesium from fertilizer, even while limiting iron uptake, which may help manage astronaut iron overload. Thirdly, in plant growth, results were substrate dependent. In perlite vermiculite, melanin plants grew larger and more water efficient, though some stress symptoms appeared. In arsenite, plants remained smaller, but melanin still reduced excess iron and supported more balanced nutrition. Across both substrates, melanin consistently boosted plant potassium, magnesium, and calcium while lowering iron levels, which is an ideal shift for astronaut diets. One exception was magnesium in melanin-treated arsenite plants, which dropped slightly due to calcium-magnesium competition within the root zone. Okay, so now let's bring everything together. On the left, we see the synthesis table. In our previous slides, I've already established that melanin improved edible biomass in PV, promoted compactness in ARC, and may had indirectly reduced plant health in PV due to abiotic stresses, and increased nutrient contents in both PV and ARC plants. To further align with NASA's crop selection priority, plant growth parameters and nutrient compositions were scored using a normalized weight score system. For volume and iron content, the normalized values were calculated as inverted. Plant diets were deficient in potassium, so it is weighted double as edible biomass. The results show that ARC substrate treated with melanin ranked number 1 and PV with melanin ranked number 2. Moreover, the stat graph shows that melanin clearly boosted water use efficiency in PV, where plants converted water into biomass and volume more effectively. In short, melanin reshaped root zone chemistry in different ways. In perlite vermiculite, bigger, more water-efficient plants through nutrient uptake lag behind release. And in arsenite, compact plants, but with better nutrient balance and less iron stress. This shows that melanin's potential as a multifunctional amendment, regulating nutrients, buffering chemistry, and improving plant resilience, which are all critical for closed-loop food systems in space. To conclude, melanin could support 3 NASA priorities: nutritional safety by reducing iron overload, substrate sustainability in constrained habitats, and water resource efficiency through moisture retention. This project is the first on trial using melanin in plant substrates, and future steps include gene expression, microbiome studies, and skill testing. Thank you for listening, and I am happy to take questions at this time.
Well, thank you very much, Margaret. We appreciate that presentation. For our participants or our attendees, please submit your questions in the Q&A tab below. Margaret, I know that the motivation for this was for crew exploration, but are there any takeaways from your study that I could go and use in my backyard, today? Is there any lessons learned from that?
Hey, that's a great question. I think that melanin is really interesting because it seems to buffer the pH as well as the cation exchange capacity. So that means like the nutrients or the roots and the soil will be able to retain more nutrients. So maybe if you add like melanin in some sort of substrate, whatever you're going to be using in your backyard, that it may help increase the nutrients in whatever plants that you're growing. If you have a favorite plant.
Okay. This was probably in the presentation, but I might have missed it. How much by weight is the melanin, compared to the other constituents in the soil?
Right. So the weight of the substrate really depends. So arsenite is a heavier substrate, and then perlite vermiculite is a lighter substrate. And I think in comparison, like the melanin amount was very low. It was only 0.25 grams used. So it's not like a 1-to-1 ratio.
Okay. Question, what is the source of melanin used?
Yeah, so the melanin that we used was yeast derived. It's made by Melitech, which is a company from Dr. Ramondes Cordero, who's from Johns Hopkins University. And I think the information of how it's made is private, but I can tell you that it's made from yeast.
Okay. Is the melanin consumed in this, or is it entirely recyclable? Could you reuse the substrates, with additional nutrients added or things like that?
It's a good question. So the method that we used to add the melanin was that we had the substrate and then we just mix it within each of the substrate types and then used it for the crop trial. I will say though that in general, like the substrate is not reused when we finish a crop trial, but that would be an interesting future study to see if like we can reuse the substrate that was used in a previous experiment.
Jim?
Yeah, just a quick question. So, have you explored duration? Like how long this could, last for longer duration missions, for example?
So the crop trial that we used was 42 days, which is like a usual crop trial for extra depth pak choi. I think that what we found was that for perlite vermiculite, it may be better for short-term duration missions because like the biomass increased a lot more in a shorter amount of time. But for long-term duration missions, then arsenite plus melanin would be better because you could have more nutrient-dense plants. So I think to answer your question in terms of time, we would want to see like for arsenite, we would do like a longer crop trial, and for perlite vermiculite, a shorter one.
All right. Last question. Maybe second to last question. What is the wavelength of the grow lights, and can you comment on light and day cycles?
Yeah, so for our trial, we used the NASA simulated growth chamber. And then the wavelength of the light that we used, well, the light intensity was, I believe, 256 PAR. And then the light intensity depended on like, it had white, red, green, and blue LED lights.
All right. Last question. Why not use synthetic chelators instead of melanin?
Yeah, so that's an interesting question. I think we know already that like natural derived melanin can be helpful in space. It has been used to protect materials from radiation. So because we already know that fact, we thought it would be interesting to see if it would be good for plants as well.
Okay. Well, thank you very much, Margaret. We appreciate
Thank you.
your presentation and you taking the time to answer our questions. With that concludes both this session and the totality of our technical sessions this week. Before I kick it over to Jim, I just want to thank Jim Wei and AAS for leading this effort in although we were disappointed that we weren't able to be in person and have ISSRDC all together. We very much appreciate here at the ISS National Lab the interchange of ideas, sharing results, and this is a great way to do that. So thank you to Jim and thank you to all of our speakers.
Ryan, no, thank you guys. And yes, thank you to all the participants. We know it was a challenging and sometimes confusing process to get here, but we made it. And so thank you all for your patience and understanding and putting this together. Again, for everybody online, all of this will be available online for quite a while. We have recorded all of these sessions. They will be posted as soon as feasible, probably in the next couple of weeks. And let's see. And another thing we're exploring is just ways to continue the conversation. So to allow more Q&A of the presenters and things like that. So we'll find out some more details on that and get back to everyone who registered for these webinars and give you some more information on that going forward. So with that, again, we hope to see a lot of you at future events. AAS has several events coming up, one at the end of October down in Huntsville, focused on human space exploration, called the Von Braun Space Exploration Symposium. And then I know that CASIS, ISS National Labs, will be participating, and likely AAS will be participating in AIAA Ascend in May in 2026 in the Washington, D.C. So with that, thanks everybody so much for being here. Thanks for participating. Take care. Have a great day and talk soon. Thanks.